Phil Ash from GRIN2B Foundation, and thank you for joining our GRIN2B Hive Webinar Week. This webinar was the first in our Research Corner series, and we are pleased to welcome Dr. Caitlin Hudac from the University of Alabama. Dr. Hudac is here to provide updates on her 2019 grant, Linking Brain and Behavior, a GRIN2B biomarker. Like many things, Dr. Hudac's grant has been at the mercy of the COVID-19 pandemic. GRIN2B Foundation approved Dr. Hudac's grant in the fall of 2019. The grant was to fund her research project on identifying potential GRIN2B biomarkers via in-person EEG testing. The pandemic forced Dr. Hudac to postpone her project during all of 2020. Now, she's finally ready to resume her project this fall. This presentation will detail the various components of her study. You will find out how you can sign up to have your bee participate in this exciting study to determine if common GRIN2B phenotypes can be detected via EEGs.
Hi, everyone. I am Dr. Caitlin Hudac, and I run the Brain Research Across Development Lab, which we call the BRAD Lab at the University of Alabama. And I am here today to give you a little update from where we are. And before I get too far, I want to make sure that I acknowledge the land that we stand on. So I reside, our lab is in Alabama, in Tuscaloosa, which is the home of the Muskogee or Creek Indian, although I am currently located in Seattle, Washington, which is a really interesting mixture of many different tribes that are more colloquially known as the Coast Salish and have over 24 distinct languages. So I appreciate the land that we reside on. So my agenda today is to give you a couple different overviews of where we've been, where we are, and where we're going. So I'm going to start by talking a little bit about why we approach the development of what we'll call biomarkers for GRIN2B by looking at work that's established some biomarkers in autism. And then we'll give you a very brief overview of what we've basically been doing since COVID shut us down for actual research testing. And then I'll spend the last part of the time really talking about what data collection will look like, which is happening this fall. So when we think about what biomarkers are, so these are biological indicators that we're trying to capture in our kiddos of all ages. And it's really to try to understand how different presentation, so when one kid looks a little different from another kid, how they might actually be linked into these different groups. And so I come at this from looking at all of these different levels of the phenotype. So I'm very interested in not just kind of the clinical diagnoses or just the existence of a genetic mutation or variant of interest, but really thinking through each of these different levels and how they connect. There's actually been a lot of work in autism specifically to kind of develop biomarkers. So there's this great program called the ABCCT, which uses EEG and eye tracking. And I was very involved in this when I was at the University of Washington. And kind of the goal here is to start and say, okay, well, if we have children with autism, we've had, over 60 years of genetic discovery, which have kind of really told us that there's a genetic cause that accounts for about 20 to 30% of neurodevelopmental disorders, including autism and intellectual disability. And so when you look at kind of some of these different presentations and you start at the gene level, we're actually able to then really kind of map onto these really discrete mechanisms. So kind of what's happening in the middle here. So biologically, what is happening in the brain specifically. So for instance, 16p11.2 has these copy number variations of deletions and duplications that have a really particular mechanism related to what proteins are produced. With something called CHD8, which is another genetic mutation that we see associated with autism, we see that this actually is heavily involved in kind of controlling for different regulatory networks. And when we look at SCN2A, as you are very well aware, hopefully by now, if not, I'm happy to spend more time talking about too. This is really kind of something that's hitting at the synaptic level and early development specifically. So we can kind of look at these different levels across our biology and understand how these might map onto really distinct individual groups. And from there, that's how we develop treatments that are really going to work because they're really entrenched in that biological mechanism. So historically, and I won't go too much into this, but our lab is really focused on looking at a lot of different kinds of genetic variants. So we've done a lot of work in CHD8 and SCN2A as well, and we'll be seeing some SCN2A families this summer and fall. And so really what we've been missing is understanding more about the GRIN2B link. And so we know that it's associated with autism, but it's wasn't immediately clear to us exactly how many children with GRIN2B mutations really are showcasing AC symptoms, because it's not necessarily the case that all kids are given a diagnosis. Not all kids might look like autism. So given that we know that it is an AC linked gene, we wanted to spend a little bit of time looking at what those AC symptoms actually look like in our kiddos with GRIN2B. So we started with a data set that involved around 35 individuals, and mostly youth with GRIN2B mutations. And we were comparing their autism phenotypes to other youth that either had a different pathogenic mutation or had autism, a diagnosis of autism. That's why they came to us. And we split that group into individuals with a low nonverbal IQ and an average nonverbal IQ. And that's because we've seen so many kids with pathogenic mutations, we're able to really match these two groups so that we're looking at both age and sex and cognition matched kiddos with pathogenic mutations. We have a lot more younger individuals with GRIN2B, so we weren't entirely able to kind of make age-related changes, which is definitely a limitation of this work today, and something that I'm happy to always kind of talk about more later on too. So in this paper, which is currently under review, we were looking at different levels of symptoms. So here's a very complex graph, which I'll walk through in a minute here, that's looking at parent-reported endorsement of autism symptoms. And so if you get to this red line, that basically is indicating that all the parents we talked to had some sort of problem on these different levels. And so each of these different kind of lines coming into the center relate to a different question on this interview. And so if there's no problems in the group, you would be down here in this little green circle. And if half of the individuals are talking about problems, they would be hitting this middle point. So it's a very complex graph. So I just want to point out a couple of really interesting things that we found from the parent interview. So first of all, we saw a lot of strengths. So one of the things that you can kind of see, so I've marked the GRIN2B kiddos in green here. You see how a lot of them are really close to the center. That's indicating that on one of our classic autism assessments, many of the parents, and in fact, in many cases, most of the parents are indicating that these children do not have a problem related to autism. So specifically, we saw fewer problems related to some of the sensory stimuli that in autism. And we saw fewer problems, particularly with how appropriate social engagement is. So the children are really, actually, I'm sorry, this is a typo. My bad. I apologize. This is social approach. So children with GRIN2B, we're very excited to approach other people. And I know that's a typo because we do see from a clinician observed behaviors that the responses are often inappropriate. So we have this kind of interesting contrast in the social behaviors where our kiddos with GRIN2B are really excited to kind of engage with other people. And so this social approach, I'm really sorry for the typo, is not something that we're seeing in these kids, unlike our kids with autism, but how they then handle those social interactions are not always quite appropriate. We also see some other kind of communication issues, but for the most part, we saw many, many strengths in GRIN2B. So just to kind of hammer home the fact that this social approach and what's known here in this graph as social motivation is really a unique strength in GRIN2B. So the higher you are on this measure, which is tracking social responsiveness, the more problems you have. And our GRIN2B kids are even threshold. So this little dash line here indicates when we might start to be worried about social motivation. So we're really seeing this as a unique strength. So that's kind of the take-home message from this work that we put together. And it kind of gives us this idea that even if we are going to be thinking about GRIN2B as an autism-related gene, we need to be keeping our kind of finger on the fact that of these social strengths, but maybe identifying and using that information about some of the problems with social responses to guide our treatment ideas. So, but many of you probably know by now that I am a neuroscientist, so I think a lot about the brain. So really what we're trying to do moving forward is to kind of take this information and understand how those biomarkers that have been developed, looking at kind of this information processing level and this physiology level, how that's going to relate to GRIN2B, given the fact that their autism profile doesn't quite look like classic autism. So we have, and I think I've presented some of this work previously, looking at some of these biomarkers for these different groups. So kind of looking at mutation groups, and we see these really interesting kind of profiles. And GRIN2B has this very clear delay. So there's an atypical habituation, which just means the way in which the brain is processing this information over time is a little different. But actually, we're seeing very classic responses. It just might take a little bit longer in GRIN2B. So that's something that we're hoping to develop and continue to push on. So when we think about kind of where we are in biomarker land, so we spend a lot of time looking at kind of the basic science mechanism and trying to then translate that to what we see at the scalp, really, with our EEG. And I'll talk about that in a minute here. So this brief mention of our future goals is really to kind of we're continuing to look for additional funding to continue this work. And it's really to kind of focus on this section here, where we have a couple different targeted biomarkers we want to test using both EEG and eye tracking, and understand how specific they might be to autism, but then also really look to see how these are really unique in these different genetic etiology groups, both at one time point, but then also about a month and a half later. So kind of looking at that change over time. So, and I'll try to be brief. I know I'm already getting a little long-winded, but our current efforts are really to actually start this data collection. We've been given the green light by our university to re-engage with off-campus testing. So we're going to be, this is me in my little car, traveling across the country. And to give you a little preview, this is, there's still a lot to kind of work out and finagle, but this is kind of the trip that we're hoping to take. And you can see some dates in mind. I have some kind of personal things that constrain me to my hometown in Mars, for example. And you can see, a nice little southern trip. And we're going to have to fly to Tampa and Chicago at some point. So we'll actually be in Chicago twice. I know there's a lot of you there. So what to kind of know what the visits are going to be like. So for people where we're going to set up at your home, usually it takes me about 45 minutes or so, maybe an hour, to just set up the equipment, because I want to make sure everything's working correctly. And there's just a lot of things to unpack and put together. And then it should be about an hour for the EEG testing. And I'm including kind of spending some time in interacting and acclimating to what this looks like. We don't need to rush anything. And so we want to make sure that this is as successful as possible. So in terms of kind of acclimating to the setup, putting the net on, and then starting the testing, the actual testing should be about 20 minutes, but I'm giving myself just an hour buffer in case we need a little bit more time for those other steps. And then I should be able to tear everything down in about 30 minutes. So we're looking at about a 3-hour block of time to reserve. In contrast, if we were doing a hotel visit, you don't have to worry about any of the setup or tear down. So you're welcome to, and this is something I'll talk about with each family about their preference. So you're welcome to kind of come to, usually I'll find a hotel suite so that we have enough room, and I'll have everything set up before you come, so that just kind of reduces some of that time. And then in addition to the EEG, we have a bunch of forms that parents are going to complete, and these are going to be online. So you are welcome to do them before or after the visit. I can even bring paper copies if you'd prefer to do them while I'm there. So for people who haven't seen what an EEG looks like, we use this high density net, which just means we have a lot of different channels that can go on the head very, very quickly. So much of my work in grad school is working with infants and other kiddos that had maybe limited verbal abilities. So we have a lot of different strategies for kind of making sure that they're comfortable. So to prepare for these upcoming home visits, what we're going to do this summer is have a plan with each family. And this will probably involve about a 10 to 20 minute phone call. And you can see some of the sample questions below just to give you a little bit of a sense of what kind of information we're looking for. We're really just trying to get to know what your child might need. Since we have such limited time with each family, this will give us that chance to really make sure that things are as successful as they can be. And through this, we'll also then be able to develop really personalized social stories that look something like this. So we can change out the pictures and really kind of walk through what each child might expect. And this will be something that you can have and share and kind of review in maybe the week or so leading up to my visit. And I'd be happy to also kind of do anything virtually that you think might be helpful if you want to kind of check in by Zoom or even FaceTime, just so that your child has some familiarity. We'll basically develop these plans over the summer, knowing what's worked well for your child in the past. So the other thing that we can do, particularly if your child has any sensory sensitivities around their head or having wet things on their hair, we have developed some EEG home training that you can do to just kind of practice what this is going to look like. The very, very basic version is just a wig cap that we can send with kind of a list of activities to do. And this just gets the child used to having something more constraining on their head that sits a little bit more tightly than maybe a knit cap would, for example. And for kiddos that might have more issues with some of the tactile issues, so with the long cords that hang down, we can also manufacture this more complex simulated cap that has buttons and pom-poms to kind of represent the different channels all across the net. And we actually even have some training procedures where there's some suggestions where you can hang progressively heavier things from the back of it to kind of even simulate that feeling of it being a little bit more of a pull. So just to kind of wrap up, we're really excited by that opportunity to kind of really think through this autism link to GRIN2B. And it's really helpful for us to know that it looks very different or at least unique in GRIN2B, so that we're not kind of blindly approaching this with an autism biomarker in mind. And we're really excited about getting to do some data collection and use our current funding from GRIN2B Foundation and our future funding that we're hopefully we'll hear about soon to kind of continue this work. And one of those things that I might have undersold, but I really think that we need to be capturing some of this developmental picture. So the future work where we're going to see each child twice, I think is going to be very, very helpful in understanding how stable these signals are over time, particularly if we're going to address this gap and think through what does our brain response connect to in terms of this these other aspects of the biology, because that's really where we're going to start thinking about potential pharmacological interventions and treatment options. So thank you for letting me share a little update from our lab. Feel free to contact me here or visit our website. And I'm excited to answer any questions.
Thank you, Dr. Hudac, for that presentation. We really appreciate you taking the time to do that. I know our families really appreciate just being able to kind of see all of this, and COVID really threw a wrench in this whole thing, and it feels like it's been such a long time since this was greenlit, and now we're just kind of getting started, but we're excited to finally get started. So my first question is are families still able to sign up
Yes. So if you are in, even if you're in an area where you didn't see me driving through on the map, let's say, reach out to me and we'll figure this out. So part of my hope is that even if it doesn't work for this road trip, I have some time and funding potentially left over at the end to potentially travel to some of the maybe less connected areas, geographic areas. The other piece I'll mention, I didn't include there's some Canadian families that are very interested. And at this moment, the borders aren't open, but if in the midst of the fall that does open, I have some contingencies for being able to come up and visit with the Canadian families as well. The other piece that gets asked a lot because we're such a amazing growing international family is when do I go abroad? Well, that's part of the reason why I was applying for other funding opportunities because there's a lot of families, particularly in different areas of Europe and the Middle East, that were very interested. So if I can just gather those funds, or even if there's enough left over here and borders are open and safe, I will be coming to you. But let me know if you're interested, and I'll keep everyone posted as some of those opportunities become actionable.
Great. Thank you very much. Last question, and you already kind of did it, but I just want to make sure I'm going to ask this question and then I'm going to sort of tell you what I think I heard. And then if you could sort of say it again, that'd be great. So essentially in outlining the long-term benefits of this study, it sounds like what I'm hearing you say is that if we can pinpoint consistent biomarkers over time from various GRIN2B patients, that will eventually lead to pharmacological treatments for consistent symptoms or phenotypes. Is that accurate? That's the goal here?
Yeah. So one of the things that I've been really excited about, and I've gotten more involved with the Cure GRIN, with the more broad category, is just learning more about really what these NMDA receptors are doing and how that might translate to what we see. And, so I'm really trying to use the brain biomarkers as a link between what your child does on the daily basis, right? So kind of what behaviors are improving, what interventions might work clinically, to understanding how those biological mechanisms may be impacted by specific variants and specific mutations, and understanding then, well, is there pharmacological treatment that might also help? So it's kind of, I see this as multifold. So in one way, I am trying to kind of help bridge between that phenotype of what you see in your child to what their biology is kind of how their biology might be constraining some of these behaviors. And just to kind of understand that mechanism. But that's really important to me because I think that these could be very helpful for clinical trial interventions. So I see a lot of this work, we call it preclinical trial. And in that little like wheel chart that I drew, that's why I put myself kind of at the beginning. And what I actually work very closely with a lot of people that are doing clinical interventions. And I have a brand new colleague who will be starting in the fall who does implementation science, which is really like, well, how well is this treatment working? And that treatment could be behavioral training, it could be cognitive training, it could be a drug study. And the goal would be to have an understanding of what GRIN2B looks like as GRIN2B, and then track if there's improvements because of those specific features. And that's why for me, the developmental piece is so important, because you need to be able to have something that is going to be fairly stable across development. And puberty can be a whole other kind of challenge just to address, because there's a lot of biological changes that are happening in a very truncated amount of time. So that's partly why we're going to try. And even I'd love to be able to do these road trips every year, right? Like that's what we're trying to do, so that, we're developing relationships with all of you, and we're seeing your child, and we're learning about the strengths and what's working, what isn't working, so that we can then map back onto what we're seeing is changing at the brain level. So I'm hoping that this is a long-term partnership with everyone, because we really want to be helping find those solutions.
Great, thank you.
Of course.
And I guess the last question then so you mentioned the idea of following up, which I agree, I think is really important to see how things change over time. And I believe you'd mentioned like 6 months, which is great. Is there any particular reason why you chose that specific time period?
Yeah, I think it was 6 weeks. I could actually be wrong.
6 weeks, I apologize. Yeah, it was 6 weeks. No.
So there's like two different ways in which we would want to follow up. Like, one is to understand how children are changing over time, just because we're all humans that evolve and change and learn and grow, and treatments work, or there's regressions that we should know about. And for those, we tend to look out maybe every 6 months or every year would be appropriate. When you're trying to decide if the brain state that you measured is fairly stable, like, you want it to be a shorter time period. So that's why that grant would be looking at like a 6-week turnaround, because the goal of that grant will be to validate these markers. And the other thing I didn't have enough time to really get into is, I spent a lot of time talking about GRIN2B, SCN2A, CHD8, and DYRK1A, because we've seen a lot of these kiddos. And it's really, really helpful for us to know what is unique to GRIN2B and how do they stand apart. And that's why I think some of that early work that we did looking at these auditory biomarkers is just so helpful, because we saw different profiles. We see how these children are different, which indicate that there's probably different avenues of intervention that are going to be necessary.
That's great. It's very exciting. Again, speaking only for myself, my own family, I always feel like my daughter is, she's different. There is no classic show, with my daughter. And so that's why I'm really excited about this work, because I think there is something unique, with a GRIN2B neurodevelopmental disorder, that puts it a little bit apart. And, some of the things you outlined in the presentation, I was like, oh yeah, that makes total sense. That sounds kind of like my daughter, which is great. So I want to thank you one more time. Thank you very much for answering these questions and for reviewing your work with us. And we're super excited that you're finally going to get started. Fingers crossed.
Me too. Oh my gosh. I'm so excited to meet everyone.
Likewise, it'd be great to meet you in person as well. So Dr. Hudac, thank you very much for your time, and we look forward to seeing you soon.
Thank you so much.